Welcome to the world of chemistry, where innovation fuels progress, here in Hyderabad. Pharma capital of the world lies a hub of scientific excellence, CSIR-IICT, Indian Institute of Chemical Technology, established in 1944. The IICT is a premier research institution in India dedicated to pushing the boundaries of chemical sciences and technology.
IICT is Indian Institute of Chemical Technology. Basically, we would like to see our institute giving solutions to anything related to chemistry or chemical technology. Of course, we broadly work on, there are 4 areas. One is healthcare, another one is chemicals, any chemicals. So another one is energy, another one is environment. So these 4 areas broadly we focus. Of course, we have multiple departments like organic synthesis, there's a process chemistry, we have agrochemicals, we have functional materials, we have catalysis, we have applied biology, we have different departments. Another one is analytical structural chemistry, that is another biggest department, so well known in the country. So like that we have multiple departments, but our overall we work on these 4 major areas. One is healthcare, chemicals, environment and energy. So that's our focus. Of course, we transport several technologies to industries and mainly process development of agrochemicals and APIs, and of course a lot of solutions, energy solutions, waste to wealth creations. These are some of the things we continuously doing, and very recent one is biodegradable plastics. So that's again very recently we transferred. So we have been continuously helping society and also industry.
I am Dr. Venkat Mohan, working as Chief Scientist in Department of Energy and Environmental Engineering. So our department basically focuses on advanced wastewater treatment as well as circular economy. In this direction, we focus on using waste as a feedstock and try to convert into valuable added products. Starting from methane by using solid waste or liquid waste, we try to look into waste as a potential and sustainable feedstock for production of biofuels and chemicals. In this direction, we are developing various processes which I would like to show you in the lab right now. This is one of the treatment system where wetland plants are used to treat the domestic wastewater or a specialized wastewater, where 3 types of plants will be used to give certain conditions that can go with treatment of that pollutants. This is the biohydrogen pilot plant which we have established with funding of Ministry of New and Renewable Energy apart from CSIR. The basic scope of this work is to look into hydrogen production from biological waste by using a specific group of bacteria, that is acetogenic bacteria. So IICT is working since last 15 years and established a pilot plant which can produce about 50,000 liters per day of hydrogen from food waste. So this pilot plant has a very unique features where the pre-treatment of waste as well as pre-treatment of culture, bacterial culture, which will be enriched for acetogenic bacteria, will be done in the system. The 10 meter cube of bioreactor capacity with all safety measures. Cascading of different products starting from methane, microalgae lipid production, proteins as well as clean water from wetlands, and this water will be recirculated. So it's a comprehensive system, it's a closed loop system where we can produce different kinds of chemicals and materials by maximizing the resource recovery. That's the concept of circular economy. We try to integrate with biohydrogen production process. As I told, we are also working with CO2. CO2 is our major feedstock to produce chemicals as well as biofuels. This is a setup, pilot plant setup, which we have established with funding of Department of Biotechnology to convert CO2, gaseous emissions of CO2, to some platform chemicals such as acetic acid as well as ethanol. The basic scope of this work is to establish a electro-fermentation strategy wherein the electron flux will be used to convert CO2 without the help of hydrogen. Basically, to convert CO2, we required hydrogen as electron donor, but in this process, we have made a system with a specific group of homoestrogenic bacteria to take CO2 and make it into value-added products. So this is a very specialized system which was recently patented, having a hybrid fermentation strategy. This is the pilot plant with a 30-liter capacity, hydrothermal liquefaction. This system will take solid waste, basically biogenic waste, and try to convert into, under supercritical conditions, convert into liquid as well as gases, apart from charcoal. So it's a kind of simulating what has happened on Mother Earth since billion of years. The biomass conditions will be creating with a high pressurized system, so that it will be converted into crude that can be subsequently make into different kind of platform chemicals. These are the reactors which we are using with electrochemical mechanism for production of various chemicals. So apart from that, we also look lignocellulose biomass as an important feedstock to convert into chemicals that has very significant industrial importance. So this is from finger millet, where we try to convert into lignin, cellulose and hemicellulose, and cellulose will be converted further into microcrystalline cellulose, nanocrystalline cellulose, and then that has a wider application in many industries, food as well as pharmaceutical industries. At present, they are trying to look cellulose as a major component to convert into cellulose acetate, cellulose bistrate, which are high-end chemicals which has lot of industrial application. This is a pilot plant wastewater treatment system designed and patented by IICT, which is being constructed in a effluent treatment plant which is developed by IICT. The basic mechanism here again using the electrocatalytic mechanism, try to use electrode assembly to flux the electrons in a defined circuit so that can function on the complex wastewater. So this pilot plant is about 100-liter capacity, which is treating textile dye wastewater. Because of these interventions, electrode interventions, the system is able to reduce complex chemicals more effectively.
Here we are maintaining the in-house culture bank. Actually, we are maintaining algal cultures, cryopreservation, bacterial cultures, fungal cultures in cryopreservation. Around 150 identified strains we isolated from wastewater and different lakes. We are collecting the samples and we are isolating the microbial strains. Then we identified as a 16S gene sequencing and we are protecting in cryopreservation technique. Sir, lignocellulosic biomass from agricultural biowaste, sir. We are collecting, and here after harvesting the biomass, then we are doing the alpha cellulose, microcrystalline cellulose, lignin, nanosilica, all these products we are from agricultural biomass to valorization of the products we are doing, sir. Actually, here this is alpha cellulose This is microcrystalline cellulose and nanocrystalline cellulose. This is the cost also, it is increasing, sir. 1 kilo of alpha cellulose is 1000 rupees and microcrystalline cellulose is around 5,000 rupees and nanocrystalline cellulose around 10,000 to 12,000 rupees. That cost also increasing while doing the processing, and we are fine particles. It has a lot of commercial values, sir. Similarly, nanosilica also, it has commercial value. By using the biorefinery process, we are extracting all other value added products from agricultural biomass.
So this is an environmental microbiology lab where we try to use microbial techniques to understand biological process, either for wastewater treatment or for production of bio-based chemicals. Apart from that, we also do microbial diversity studies. Recently, we also started working on wastewater-based epidemiology since COVID season. So we look into the waste streams and try to collect samples, and based on the RNA, we try to quantify the load of COVID initially, and subsequently at present we are working with antimicrobial resistance in community where based on wastewater only. So we are at present testing about 128 genes apart from 10 mobile genetic elements. So this wastewater based epidemiology is very much required at present, particularly with respect to antibiotic resistance, to understand its prevalence in a community that can help to give a kind of strategies to medicinal practitioners. So as part of bioelectrocatalytic systems where we are applying for different strategies, this is one area where we are looking into plant growth by taking advantage of plant roots and microorganisms, that rhizosphere, wherein microbial metabolism will give the electron flux. So we try to keep the electrode assembly in a defined way at root zone and try to flux the electrons so that a short electric field, electric shock will create. So that has a significant influence on plant growth, where you can see the experiments by inducing self-induced potential to the system. The growth of plants as well as CO2 sequestration can be increased. Number of plants, number of pods, number of leaves, area of leaves can be increased by a simple strategy by keeping some kind of conductive material at rhizosphere and then diverting electrons flow in a circuit.
I am Dr. Srihari, working as Chief Scientist in the Department of Organic Synthesis and Process Chemistry. So our department has over 19 scientists and a technical staff 10 members, and we have a vast driving force for us, that is by 100 PhD research scholars who are here for doing their PhD and will be doing for 5 years of course of time. And we also help dissertation students who will be working here for 6 months, and we have over almost 50 students working for their dissertations. In this department, we work on focusing of total synthesis of natural products and complex bioactive natural products, also synthetic molecules. And very recently, our department has been engaged in process development for several key intermediates that are used for the API synthesis. We work on method developments where we found CC bond formation reactions that are used for total synthesis of complex natural products and also bioactive molecules. So we try to use several reagents such as organocatalyst or metal-based reagents and try to develop atom economy procedures for making the key intermediate synthesis, and we also try to develop processes for lengthy molecules which are synthesized in long steps.
My name is Ruchi. I am working as a PhD student in our OSPC department. My main area of research is flow chemistry, which is related to like photochemistry and electrochemistry we are doing.
Yeah, I am P. Sivaram Krishna. I am working as a UGC Senior Research Fellow, working in the Department of Organic Synthesis and Process Chemistry. My group is working on total synthesis of natural products.
Here, this is a National Mole Bank, and the capacity of National Mole Bank is 1.6 million samples we can store here. This is at -20 temperature under pure nitrogen. It is a inert conditions where the synthetic and natural product compounds are safe without changing physical and chemical properties. We are the first in India and South Asia also. Storing of a compound, see major compounds we will receive in these vials, which is having a barcoded, and this is a vial barcode, and we have array barcode, and we have tray barcode. We have 3 barcodes. For one compound, we have 3 barcodes. Each array contains 120 plus 120, 240 compounds we can store one attempt. Now we can see the whole vials barcode. We can take the picture from IO station. Now sliding door is lifted. The tray is slowly moving inside the universal store. Now you can see the tray is slowly moving inside. So after coming here, we will send it to end user. Depends on their compound activity, they can go for a publication, or if it is compound is good activity, they can file a patent, or depends on compound activity, they can send their technology to the industry people and make it bulk level production and reach to the common public in affordable price. That is the main motto of the National Mole Bank. So presently we stored 1 lakh samples we can store here.
Hi, I am Dr. Krishnaya, chairing this Fluoro-Agrochemicals department at IICT. This Fluoro-Agrochemicals department, in short we call it as FAC, where in this department we have 3 domains. One is agrochemicals work, and other one is fluorochemicals work, where we are presently standing here in the fluorochemical department, and the other wing what we are working is the semiochemicals. So these are our main activities which goes into the industry, and you can also these the final finished products we store in these kind of cylinders. So very carefully our colleagues will conduct the experiment. The final gases, they will be stored in these kind of cylinders, and we show it to the industry.
This is Dr. Lingaya. I am heading the Department of Catalysis and Fine Chemicals. In this department, we mainly work on the development of the catalytic processes. The catalysis is the area which has wide applications in the sectors like energy, environment, and in green chemistry. Here we have different techniques which can be used to know the catalytic properties like its surface area, its acidity or basicity, its pore structure, crystalline nature, the particle size, either it has any nanoparticles or clusters, all those things one has to know. Then once we know those things only, then we can understand its activity. So this is the where we are do the characterization using the different techniques, like it is a surface nitrogen surface absorption analysis, and this is the where we use the chemisorption to know the acidity, basicity, and also we have IR and other techniques to know the stability and other things. Here we have a facility where we can do the reaction, and during the reaction itself we can understand the nature of the catalyst and the reaction intermediate forming on the surface of the catalyst. So this is a very important characterization techniques used widely for the characterization of the catalyst. In this department, we have about 15 scientists and equal number of technical assistants and technical officers. In this department, we mainly work on the development of green catalytic process, mostly useful for the industry, and also we work on energy and environmental related research. After preparing the catalyst and characterization, the main aspect is the evaluation of its activity. So generally the catalyst that what we prepared will be tested in this type of reactor. So this is for example is a continuous vapor phase reaction. This is where you can make the products continuously. That means we take the catalyst in the middle of the reactor looks like this, and in this we will take the catalyst, and then the reactants will be flow and reacted at a given temperature, and the products will come, and then we will analyze the products, and that like this we evaluate, and depending upon the activity, and the activity is measured by the gas chromatograph or other analytical methods. Then we already know the characteristics of the catalyst, and now we know the activity of the catalyst. Then we will understand why this catalyst is giving high activity or why it is not giving activity. All these studies we do in this type of reactions. Here for example we are doing one reaction where the carbon dioxide is converted into synthesis gas, which can be used for the preparation of CO and hydrogen, and that can be used for the preparation of methanol. So CO2, now a lot of research we are doing on the conversion of the greenhouse gas CO2 and methane into valuable products. That is the one of the active research is going on in our department. This is fluidized bed reactor because in this case the catalyst is in the fluid form, and these reactions are used particularly for the exothermic reactions, and where the catalyst is always in the moving mode, and then it will we adjust the reaction temperature and we control, and once the reactants, the products will come out, and we collect here and analyze by the gas chromatograph, And this is a continuous automatic system where we control all the parameters through PC. So these are the other reactors here where we do the bench and pilot scale studies. Once the catalyst we tested in the laboratory, then if the selected catalyst we will evaluate at higher scale, and most of the industry sponsored project we do research in these reactors. Yeah, this is one more reactor where we are particularly working on the conversion of CO2 into synthesis gas. This is called CO2 methane bioreforming, where we do the reaction around 800 degrees centigrade and continuously we analyze by the gas chromatograph. So this is one of the recent facility in our department for the evaluation of the catalysts.
Hello, I am Dr. A.G. Rao. I am the Chair of Energy and Environmental Engineering Division at CSIR-IICT Hyderabad. Our group working in the area of multidisciplinary areas of energy and environment actually, and we also work in the interface between energy and environment. So our idea is that whatever the solid waste is generated in India from municipal urban bodies or local urban local bodies or industries converted into useful products. In that bargain, we are actually generating biogas from food waste and market and vegetable waste. And as a part of this exercise, we have installed approximately 35 plants across India for the food waste, market vegetable waste and organic fraction of MSW. All these plants are working based on a technology, AGR technology developed by CSIR-IICT.
Myself, Dr. Naveen Kumar. I work as a research associate here in this laboratory, and this is our wet lab laboratory in the department. Here we try to characterize all the characteristics of the biomass which we get from all the clients. The characteristics are as follows. We do total solids, volatile solids, VFAs and COD. Apart from that, we also do biomethanation potential test and specific methanogenic activity to understand the methane potential about the biomass and also the methane generation capability of the microorganisms.
Namaskar, this is Dr. Pradosh Chakraborty. I am heading this Department of Oils, Lipid Science and Technology at CSIR-Indian Institute of Chemical Technology. Basically, we deal with all aspects of oils and fats and its processing, its byproduct utilization, then the derivatives from oils, and say some non-edible applications like in biodiesel, in lubricant, and of course we also use these materials and get some of the components from the byproducts and utilize for gene and drug delivery. There are many projects which we are running at the present moment. We have state-of-the-art analytical facilities at the IICT, and this facility is NABL accredited. Here we actually work on gas chromatography and mass spectrometry, GCMS. These analyses are very important for oils and fats, and our research students, they are working on their research projects. Apart from that, we get hundreds of industry samples, and since it is NABL accredited, we get different samples from other industries, then government concerns, and also we are recognized as National Referral Laboratory by FSSI on oils and fats. So here, this is the heart of our department. So we work on the quality of oil we check the fatty acid composition, and then we check the antioxidants or minor components present in oils which are having nutritional qualities.
I am Dr. Shashi Kalivendi, a chief scientist and chair of the Department of Applied Biology. So basically we are a team of 21 scientists working in the areas of cancer, CNS and metabolic disorders, infectious diseases and immunology, structural and computational biology, regenerative medicine, pharmacology and toxicology. So our department main focus is in the identification of novel drug targets as well as potential small molecules for therapeutic purpose. So coming to my lab focus, we are actually more focused on neurodegenerative diseases with a particular emphasis on modulators of mitochondrial bioenergetics and protein turnover mechanisms. And soon my colleagues will be present showcasing their research activities.
Our lab mainly focuses on neurodegenerative disorders and other pathophysiologies related to that. So currently this is our cell culture, and we maintain mainly neural cell lines, and we also occasionally works on glioblastoma and other cancer cells. We work on neurobiology mainly. So this particular machine is one of the India's first machine. It is known as CSIR's extracellular flux analyzer. So what this does is, it will help you understand the bioenergetics of mitochondria. So if we have any particular compounds of treatments that affects the mitochondrial bioenergetics, we can identify at what stage the action is taking place.
I'm B. Supriya, Senior Technical Officer from Applied Biology, IICT. It's a confocal instrument with Nikon Eclipse TI model. It is used to image the cells of fluorescent dyes, tissue sections, small organelles of zebrafish and Drosophila. The specialty of this instrument is to take the orbital sections of the sample to form a 3D image.
I am Dr. Sumana Chakravarti, Senior Principal Scientist in Applied Biology, CSIR-IICT. Our lab mainly focuses on behavioral disorder, especially the sex difference. We are finding out the underlying mechanism of sex difference in mood disorders and neurological disorders. So to do that, this lab is specifically for molecular biology, but all the experiment starts with the behavioral setup. So we check our behavioral study. If they are diseased and they show some disease behavior, then we collect their brain for molecular study. We check our cellular study, brain cellular study, which protein is the marker of those neurological disorders or mood disorders, so that the chemistry people can target some protein to find out the antidepressant, better antidepressant and better anti-anxiety drugs.
Hello, I am Dr. Pragati Kishore Prasad. I am working as a DST inspired faculty in Department of Applied Biology, and this lab belongs to Dr. Anthony Adlagatta. This is a structural biology lab. They are studying the structure of protein at molecular level. So they do protein expression and purification in this lab. Once they get the proteins in hand, then they actually crystallize it and see the protein structure at molecular level. So crystallization is done here. This is X-ray diffractometer, where they actually study the protein crystal structures.
Myself Sonam Swain. I am working with Dr. Nisanjan in the field of molecular and cancer biology. In our lab, we are doing molecular biology, cell cloning, DNA cloning and transfection, all the things. Basically, our lab is focusing on protein ubiquitination, protein degradation. Basically, here we perform our Western blot techniques. First of all, we use Ponsu stain. This is a reversible stain that can bind the protein reversibly. Then we wash the blots. This is our Western blot apparatus. Here we wash the blot, we do blocking, primary antibody incubation and secondary antibody incubation. After the Western blot, we can prove our blot and we can check the expression of our protein in dark room.
Myself Dr. Anjana Devi. Our lab has been working in the area of cancer biology since 16 years. Our particular focus is on pancreatic cancer research, and we have been concentrating on autophagic mechanisms of cell death. We are also trying to understand the crosstalk between autophagy and apoptosis and exploring novel small molecules which are being synthesized at IICT for selective autophagic cell death in cancer cells. By this research, we have identified few molecules by in vitro and in vivo studies, and further validation and characterization of the mechanisms and further data is in progress. Further experiments are in process to understand and the exact mechanism of action of these molecules in vitro studies.
Hi, myself Partha Sarthi, student of Dr. Anjana Devi. My work is concentrated on role of autophagy related to ID proteins, how the ID proteins regulation will lead to autophagy and what are its downstream effects and how it is leading to cell death of the cancers.
Hi, my name is Shabarinath Chilaka. I'm a Ramalinga Swami Fellow working in IICT. We are working on epigenetic field. We are mainly focusing on the inflammation, epigenetic mechanisms of inflammation. So epigenetics is a scientific field which studies how genes are switched on and off. So most of the diseases we are having is some or other way is due to the alterations in genes.
Myself, I'm Dr. Sandeep Bharate. So I'm Senior Principal Scientist and currently heading the Department of Natural Products and Medicinal Chemistry. So our department, Natural Products and Medicinal Chemistry, we are dealing with the drug discovery, that means discovery of new molecular entities for various therapeutic indications. So in our department, we work in mainly natural product isolation. We characterize those natural products and then we check them for the biological activity so that we can pursue their medicinal chemistry towards that particular therapeutic area. Once we design the molecules, then by chemical synthesis we synthesize, make those compounds, we characterize by various spectroscopic techniques. And once those molecules are synthesized, we have to test them in the in vitro assays. So whatever the proteins we selected, then these enzyme assays are there. So for that, these kind of plates we use where we incubate the enzyme with whatever compound is synthesized, and then we detect whether compound is inhibiting that enzyme by some biochemical assays. And these readings, we use this instrument called as a multimode reader, where we can check endpoints by absorbance, fluorescence or luminescence, and by Excel or different softwares we can determine whether the enzyme levels are reduced because of the compound. So by this we can determine which compound is better and which compound can be taken further for detailed analysis in animal models or even ADME studies. So this is the initial step after synthesis of the compound to select the compound for the next stages.
So I am Dr. Haridas. I am a Senior Principal Scientist in the Department of Natural Product and Medicinal Chemistry. This is our actually medicinal chemistry lab wherein we synthesize the compound hoping to find various new inhibitors for different diseases actually. So the large amount of chemical diverse compound generally we make it in the lab, and then these are tested in collaboration with our applied biology team, and then once we find the actives actually we take it forward to the leads, and then we try to develop the lead candidates for especially we in the lab actually we work on the tuberculosis field wherein we try to find out new inhibitors which can tackle the tuberculosis. So especially in the case of tuberculosis, drug resistance is a big factor, and to tackle this there are only a few drugs available, but they are also cardiotoxic. So our main objective of the lab is to develop molecules actually, the preclinical candidate which is effective against drug resistant tuberculosis. That is the main objective.
am AVS Sharma. I am a Senior Principal Scientist and Chair of Department of Analytical and Structural Chemistry of CSIR-IICT. The department has 4 verticals, which are Center for NMR, Center for X-ray Crystallography, Center for Mass Spectrometry and Separation Sciences. The core strength of the department is expertise and scientific as well as technical staff which are involved in institute's research programs which are of integrated and independent projects. The department is a backbone of institute's research programs which caters the needs of the institute's internal research programs.
I'm Dr. Lakshmi. I'm working in the Mass Spectrometry Center. We have different types of varieties of mass spectrometers here in our section. We do analysis for various compounds which are present in environmental samples and also biological samples and in food samples. We do analysis for our internal samples received from IICT and also from the samples from various outside agencies like pharmaceutical industries and also from universities.
Yeah, good afternoon. This is Center for X-ray Crystallography and our activities involved in drug polymorphism, multi-component analysis, drug co-crystal salt formation. This is the single crystal X-ray diffractometer where you can get the 3-dimensional crystal structure, and also we have a powder X-ray diffractometer. Powder diffraction is a fingerprint of each compound. With that we can clearly differentiate the polymorphic form of all the organic compounds which are submitted. We are involved in our own in-house project as well as a lot of industries are approaching us for their polymorphic differentiation, co-crystal salt confirmation, and they want to confirm their structural ambiguity which is coming from a lot of synthetic steps.
I am Dr. Yamuna Rani, and I am leading the Chemical Engineering and Process Technology department. Our department focuses mostly towards translational research, so we take up the reactions which are developed in collaboration with other departments to the pilot scale and then to the commercial scale. And we are involved in bench scale studies, pilot scale studies and commercial scale designs, and we also handhold the industries to carry out their commercial production. So we give them guarantee runs as also the commissioning of the products.
Myself, Dr. Sumana Channa and Dr. Naveen Kumar, both of us are in charge for this pilot plant complex. CSIR-IICT has 3 pilot plants, pilot plant 1, 2 and then a hydrogenation plant, where this entire complex of pilot plants cater the needs of individual scientists across the lab as well as cater the needs of industry, which is useful for scale up and commercialization activities. So here we have several set of equipment, reactors and separation units, distillation columns and different types of reactors which can be used for these scale up activities.
So we have several units which are in operation here in this lab also. So this is a continuous reactive distillation unit which is used for reaction accompanied with separation, and it is in continuous operation. Very rarely you find continuous units in operation in labs. They are generally there only in commercial operation. This is a continuous reactive distillation unit where reaction as well as separation are taking place at the same time. So, and moreover it is in continuous operation, so which is a rare phenomenon in labs. We generally have only batch operations in labs, but this is at a commercial, which is closer to commercial scale operation. This is a high pressure fixed bed reactor. So here we conduct high pressure reactions, vapor and liquid phase reactions. And we have online measurement which is available here, which we use to monitor the reaction. So this is a nebulometer. It is used to measure the vapor liquid equilibrium, and it can be used at from atmospheric to vacuum at different vacuums. It is useful in design, in calculation of the design parameters for the distillation operations.
This is one novel combination of amine and solvent which we have identified. This we can see that upon CO2 capture, the liquid amine had precipitated into solid, where you can see that a solid precipitate which indicates the amine and the CO2 captured, and this is a lean phase indicating the solvent.
Hello everyone, this is Dr. Pavani, working as Senior Scientist in Chemical Engineering and Process Technology Department. So here we work on process development, process intensification and scale up. Basically we do lab scale process development, we take it up to pilot scale and commercial level plant design and basic design reports. Basically all the R&D works that we carry out here are based on green engineering processes, wherein sustainability is the main key.
Hello, I am Dr. Alka, currently working as a senior scientist at Chemical Engineering and Process Technology Department. So currently we are working in the area of process development, pilot scale design and commercial plant commissioning also we assist. So we are working in the area of like mechanochemical assisted process where biomass valorizations we do. From biomass different value added products we make like furanic platform chemicals we make, then biochar, bio oil kind of things we do, and then we are also working in the area of plastic depolymerization.
Hello, I am Dr. S. Sridhar, Chief Scientist from Chemical Engineering Department of Indian Institute of Chemical Technology, Hyderabad. Here I am showing this atmospheric water generator, which is very useful for water scarce regions, hilly regions and coastal regions where there is no water to treat. So we depend upon the relative humidity in the atmosphere and generate the water from the relative humidity by condensing it. And then we add minerals to the water and make it consumable for our consumption in hill stations. Apart from drinking water purification, we also have some variations such as production of alkaline ionized water from municipal water, and this is an immunity booster, and we recommend a pH of about 8.5, which is within the BIS standards of drinking water. And this alkaline ionized water is made by membrane electrolysis, and it helps to relieve hyperacidity and joint pains.
My name is Dr. Madhumala. I am working as a scientist in Chemical Engineering and Process Technology Department. We are working on membrane-based gas separation system, processes, wherein this is a gas separation unit, like mainly use it for separation of industrial gases, especially olefin paraffin separations, hydrocarbon removal and biogas upgradation, and it can also be used for carbon capture. Based on the partial difference pressure across the membrane, the separation of the gaseous species occurs through the membrane. So here we use thin film composite membranes as well as dense membranes.
Hello everyone, I am Sugari Chandrasekhar. I am Principal Scientist working in Chemical Engineering and Process Technology Department. Actually, this is Central RO plant where it is catering the water needs of entire IICT plant, and also this is having 2 water filters as well as 2 membranes. First initial water will be come to the filters, then again it will pass through the membrane. Again here we will adjust the flow rates for the water recovery as well as pressure also we will adjust, pH also we will adjust there. and then from there again it will go to the intermediate tank, where before the intermediate tank some UV chamber as well as some pre-filters will be there. From there the intermediate tank water will be coming to the product tank. Product tank and two collection points. Collection points are outside are there. From there all entire ICT staff are collecting the water.
So we are working on the process called hydrodynamic cavitation, which is basically a mechanochemical process. So this is basically intensifying the chemical reactions as well as different kind of mechanochemical effect to intensify the process. So in that aspect we are working on that one area like bio-leaching, where we are working on the electronic waste recycle. So a lot of like critical element, like its recent budget also, if you see, they have intensified on the critical element recovery or rare earth recovery from the electronic waste.
This is Dr. Rohit Kumar Rana. I am chairing the Department of Polymers and Functional Materials at CSIR-IICT. This department works on various functional materials. As the name suggests, it involves polymeric materials, coatings, paints, adhesives, materials for energy application, especially on green energy where sunlight and other resources of energy can be captured and converted into electricity. And also we work on energy storage, where our focus has been on rechargeable batteries. Now coming on to the polymeric materials, we have been working for several decades now, almost 7 decades of work on polymeric materials. Our department started with a name called paints, then surface coatings and paints, then organic coatings, then it became polymers and functional materials.
I am Dr. Rohini Kumar, working in polymer division, and we have developed a material based on N-butyl to cyanoacrylate, which is useful as an adhesive for surgical applications. And this technology has been transferred to many industries in India, and it is there in the market even now. And this is used in many surgical applications like ophthalmology, gastroenterology, and dental and maxillofacial surgeries. And it has got very good demand in India now.
This is a transmission electron microscopic instrument where it is used to see the nanoparticles which are of nanometer size, and for that we need to prepare the sample first. The sample is dispersed in ethanol and then drop casted onto a copper grid which is now loaded onto the sample holder, and this sample holder will be taken out and inserted into the equipment. This is the whole equipment called transmission electron microscope, and the sample will go into the chamber where it will be at very high vacuum, under which electrons beams will be falling on the sample, and it will be transmitted through the sample holder or the sample copper grid. So based on the contrast or the atomic density of the metal, we will be observing a 2D image of those materials on the screen. This is one of the sample where we can see sizes of 5 to 10 nanometers of gold particles deposited on zinc oxide particles. The small dark spots are actually gold particles which are 5 to 10 nanometer.
this is Dr. Muralidharan, working as senior scientist at CSIR-IICT. So basically where we are here is animal facility of IICT. It is centralized animal facility catering around 4,000 square feet. So basically this is the first animal room where we are holding the animals. Animals are housed in a caging system called individually ventilated caging systems. The individually ventilated caging system has the different cages. Each cages will serve as an individual unit. The filters are HEPA filtered and the air the animal breathe will be very clean. Also the feed we are giving to the animal is sterilized and it is free from any kind of contaminations. So particular environment is fully free from any pathogens and the HEPA filtered air circulate around 15 times per hour.
Hello everyone, I am Dr. Yogesh. I am working as veterinary pathologist at CSIR-IICT. So in our facility we have different various kind of models, animal models of mice as well as rats used for preclinical studies. So here we have different kind of models like diabetes, atherosclerosis model, various cancer models and inflammation models. So this is one of the animal holding rooms. I will show you now. So here this is a caging station where animals are observed under caging station because there will be air barrier. Inside that only we will be opening the cage so that the sterile environment will be maintained around the animal, so that will avoid contamination to the animals, so that we will get the enhanced or reproducible research results. So I'll be showing one cage. So this is cage-training station with light as well as air filter around it. So inside the cage-training station we will be opening the cage so that we can observe the animal for any clinical observations or any experimentation. So animals also should be treated in a very refined way, because animals should not be suffered in any at any point of time during their experimentation, prior to experimentation or after experimentation. So I will show how we will be handling the animals. So you can see how we are handling the animals. Animal is staying quiet and very comfortable. So with this we can observe the animals for any kind of physical examination. For identification of cages we have a cage cards, a dedicated cage card. So all the animal details will be written on this card.
In IICT we have so much facility like NMR facility, we have HPLC, we have HPLC, CLSIR in our lab, own HPLC we have, and mass spectrometry we have, and HRMS we have. If we submit the sample within 2 hours, we will get NMR data also, and there is no need to go anywhere outside. If you want any facility regarding chemicals, scheme, 2-3 scientists will come together and they will sort your problem. So if you want to do research here, it's a very good facility here, and there will not be trouble to do research here. And in short, if I want to say 4 years I spent here, and how 4 years completed, that was smoothly completed 4 years here.
I came here in IICT last year, and it's been a wonderful journey since now. And I'm looking forward to do a wonderful research, which here I can do multilinguistic research all over in this institute, and I get very helpful from this, from the scientists, from every department, and I'm looking forward to do my research, and I will try to come up with good results.
So it is the very nice place. I joined 2 years back here. So up to now I feel very comfortable and good place for work. And here we will get all facilities like for analysis also, synthesis. For everything it is the one of the best platform for doing PhD.
And we are going to be the nation by that time which are self-sustainable. I think we can make most of the things, at least if by 2047, I think we should be able to reach there, whatever our vision is there right now, our government.
CSIR-IICT, where molecules meet purpose, where science meets society, and where innovation becomes impact. For more science updates, keep watching Neo Science Hub.